All right, if you're just getting into your senior biology and you're getting into organic chemistry or biochemistry, you're going to come across a number of different looking molecules. And if you have not yet had a chance to get into organic chemistry, and this is your first dive into it, there's some things you need to know. And this is a question that comes up a lot as I teach this course. Where did the carbons go? As you start to look at certain models or structures of molecules, you will start to notice that there are different ways of writing the same molecule. And it's best to come forward with that now before you start getting into your biochemistry. So let's take an example here. This is glucose, 6 carbons, 12 hydrogens, and 6 oxygen. And you'll often see it written like this. This is like the full structural model. And you can see here they've listed all the elements, like they're all there, and you can see that they are indicated the bonds, the covalent bonds that are holding these atoms together using lines. This is your basic overall complete structure, your full structure. But you're going to start to notice that this is written in different ways. And one of the things that starts to come up is a slight variation on this. This is the hashed or the wedged notation that is being used here. And you can see it's very slight, just along that sort of bottom edge there. You can see that it starts to get thicker in that line. This is meant to give a little bit of three-dimensional perspective on your molecule. Let me give you another example here. Take a look at methane. Actually, there are 2 methanes here. They are both the same molecule, except one is drawn slightly different than the other. Now, this one over here is trying to show you that little bit of extra information here about the dimensions or the 3D shape of this molecule. In particular, if we look at these 2 bonds here, they're both covalent bonds, but this thicker lined bond here is trying to show you that this hydrogen that's bound to this carbon is actually coming up off the page or up off of your screen if you're looking at this on a device. This other bond here that's made up of these dashes or this hash marks, that's trying to show you the covalent bond that's actually behind the page. So it's trying to give you that little bit of 3D to your structure of the molecule that you're looking at there. So if we apply that to something like our glucose model, you can see here that with this, we started off with a molecule that looked like this. And now when we're looking at a molecule like this bottom edge here is basically coming up off the page. That's what they're trying to show you here. It's like a little bit of 3-dimensional information there about how this molecule might actually look. All right, so that's the first part. Again, they call that the hashed and wedged notation. Then you have something like this. So again, this is the same molecule. This is glucose. And if I go back to the previous one, you can see we had all of our atoms there. And all of a sudden, then this is where the questions come up. Where did the carbons go? This is us starting to get into more of the skeletal or the bond line type of structure where we don't necessarily have to write the carbons in. Why? Well, it's because in organic chemistry, there are so many carbons. We are basically made up of long chains of carbons. So it's implied, and the carbons can be identified where the bonds either meet or where you have these corners like that. Those are where your carbons are located, like the vertices where the bonds all meet. If you don't see an element there, it's implied that it's carbon. All right, but you can go from this to something like this. So now all of a sudden, again, back to what we had before, now all of a sudden we've lost the hydrogens. Again, getting into this skeletal type of structure or the bond line structures, the hydrogens now that are there to complete the bonds with the carbon are also implied. So now you don't have to draw the hydrogens here, and you can actually see that this is basically carbons are missing, the hydrogens are missing, or at least they look like they're missing. They're actually there. It's implied. Now at the top, you can still see that we have a carbon there, and that carbon we can actually rewrite like this. The carbon is there. It's where that little bend is. And yeah, the hydrogen's also gone from it. It's implied that it's there. You can start to see how this and this and this, they're all the same molecule. All of these are basically just different ways of drawing glucose. Now there is one more that I want to show you. This. Now this is also glucose. And you can see that we have those same types of conventions there. You can see that we have the corners or where the bonds all meet. So those are the carbons. And you can see here that we also have no hydrogens, but they're implied. So the hydrogens are there. Now, the only elements that we really do need to show are all the other elements that are involved in this molecule. So that's why you have the oxygen still there. And you can see that we have the hydrogens not bound to the carbons, but bound to the oxygen. So we have to show that as well. So why would you draw it this way? Why not just stick with something that looks like this instead? Well, that's because this is actually trying to show you the angles at which those bonds are occurring. If you ever use a molecular model kit to build glucose, you're going to notice that it doesn't really take on this nice hexagonal shape. It does have a bit of a squashed look to it, and it does have a bit of a bend there where you can see that it's not a very flat looking hexagon. This is much more accurate in terms of what you would expect to see if you were to build a model of glucose. And so that's what they're trying to show you here, is it's paying attention to the angles at which these bonds are occurring off of the carbons. So just different ways of drawing glucose. But obviously, as you go through biology, it's not just glucose you're dealing with. You're going to deal with a whole bunch of other molecules. Let's show you one here. This is a triglyceride. And just looking at this structure, you can see that we have these lines here. But now you should be able to understand that there are carbons there. Everywhere you see a corner here is a carbon. And yes, at the end here, where it ends, that is also a carbon. Now, what's attached to the carbons? Because you can't see any other elements there, it's the hydrogens that would be required in order to complete all the bonds for the carbon. So the hydrogens are there as well. Now, this, even though it does look a little weird, this is a lot faster to draw than, say, doing something like this. This is what those lines are implying. You have those carbons, you have those hydrogens there, but you can see it's quite a lot. And you would have to basically write all of these out. And that is very time consuming instead of just doing something like this. This is a small chain of amino acids. And you might not be there yet where you're talking about proteins or polypeptides, but that's not important here. What I want you to focus in on is the way that we can write this molecule. So we could also write it like this. Now, again, seeing where the corners are or the vertices are, those are where the carbons are. And anywhere that you need hydrogens to complete the bonds for the carbon, well, they are also implied there as well. So we don't have to show those. Now, everything else we do have to show, like we need to show the nitrogens, we need to show the oxygens, and we need to show the hydrogens that are connected to the nitrogens as well, because they're not connected to a carbon. And also here, I know you're going to look at the R. And what's the R all about? Well, when you start to get into proteins and looking at amino acids, this is kind of like a placeholder for the rest of the organic molecule that could be attached to the amino acid. So you'll learn about this. There are other things that you can attach to the amino acid that will define what kind of amino acid it is. But for right now, we just put an R there as a placeholder. So you have to show that bond as well. And the rest are just carbons and hydrogens that are implied in the way that we've drawn this skeletal or bond line structure. So I'm hoping that this helps clear things up for you as you start to go into other textbooks, other sources, online, written, whatever, academic papers, because you will probably see more of this type of molecule where they are using these styles of drawing your organic molecules, where you're not drawing in or labeling in all of your carbons and all of your hydrogens. It's a short form way of doing it. It's faster, it's more efficient. And yes, hopefully this helps you recognize that, because this will be a huge help for you as you start to get deeper into the course. Good luck.